Hi, Roy Trevelyan, Biology Bites. What I have here is a dialysis tubing, little plastic tubing, and I filled it with cornstarch and glucose. I've clipped it off, and I'm going to stick it in this beaker of iodine, and we'll see what happens. So what we're talking about today is a semi-permeable membrane. That's the dialysis tubing. It's got little holes in it, right? It's semi-permeable because it lets certain things in and out of the membrane. So things that are large and things that are charged have a hard time getting through membranes, generally speaking. Notice that rhymes. Large and charged molecules will have a difficult time getting through a semi-permeable membrane, or what we call differential permeable membrane. Now, when we talk about diffusion, I use the phrase high to low. What that means is molecules want to go from a high concentration to a low concentration. That's diffusion, and it's natural. It's entropy, right? I bring in a hot pizza. Before you see the pizza, you will smell the pizza. The molecules move from the pizza in a high concentration across the room and into your nose to a low concentration. That's diffusion, right? Have you ever spilled a glass of milk and it accidentally went back in the glass? Probably not. Right? Things go from high concentrations to low concentrations. When we talk about water, we're talking specifically about this process called osmosis. It's the diffusion of water through a semi-permeable membrane. This here is my semi-permeable membrane. It's that plastic tubing that I just showed you, and these are the microscopic holes that make it differentially permeable. Permeable to some things, not permeable to others. Large and charged, remember, have a hard time getting through the cell membrane. Well, if we look at my beaker here, what we have on the inside of the tubing is these big, huge polysaccharides called starch. There should be a mental picture of what a polysaccharide is, right? We don't memorize, but we internalize. By now, that should mean something to you. Big sugar. These littler sugars are glucose molecules. Chemical formula, C6. H12, O6.
Memorize.
Right? Memorize. It's the only thing we're going to memorize out of this. Take a bunch of these, you make those. On the outside of my beaker is these iodines. These AKI stand for potassium iodide. No worries. We just need the iodine, and that's the form it came in. Now, these molecules are going to want to go from high to low. The iodine molecules in the water are want to go from high to low. How are we going to know what they've done? This experiment is just qualitative. We're just going to change the colors. And when we change the color, we'll know what went where. You need to realize that when this iodine comes in contact with starch, it turns the starch purple. If you've got a cotton shirt and you spilled iodine, it'll become this dark purplish black color. So we'll know if the iodine got in because the inside of the membrane will be purple. And we'll know if the starch got out because the beaker then, the solution around the dialysis tubing, would be dark purple. I don't think the starch is going to be able to get through those pores. I think it's too big. Remember, large and charged. We'll see about the glucose molecules. And we'll know if the iodine got in because it'll turn purple. How do we know about the glucose? Well, for the glucose, I'm going to use this test strip. Similar to what you'd have if you have testing for diabetes in your urine, you'd see sugar in the content. When I put this outside this beaker, we should see glucose getting out if it was permeable to this membrane. Or in other words, are the holes in the dialysis tubing big enough to let the glucose out? How much time we got there, Ms. Tron?
4:18.
Oh, nice. That's what we're talking about today. Diffusion and osmosis and this idea of a semi-permeable membrane. This is a qualitative experiment. And what they'll ask, I've seen it anyway on the test, is they explain this and then they say, hey, how would you turn this to a quantitative experiment? And I want you to think about that. Let that roll around your head. We're going to work on not panicking, right? So the more questions I hit you with, the more you practice, the better you get at not panicking. Right? We have this little phrase, paralysis by analysis. Think too much and don't answer anything. And we can't do that. Anyway, let's take a look on how our tubing's doing. Now, I don't know if you noticed, but I did the switcheroo. I took a new tubing that was already in there, and we're starting to see a purple color, especially around the knot I tied, right? What does that tell us? If the purple got in, or if inside turned purple, and notice the outside is still brown.
Iodine got in.
Iodine got in to the bag because the starch is starting to turn purple. Did the starch get out?
No.
No. Otherwise, the iodine should have turned purple. When those two come in contact, that's how we know it what traveled where, if it turns purple. Now, I got to open this down here, and I'm going to put the test strip in. And the question is, I didn't need to. Well, we know there's glucose in there. I put it in there, right? I meant to test the beaker. We didn't really need to open that. Well, shoot.
Want me to do it?
Nah, that's all right. I'll just do the beaker. Let's assume it's in there. But if glucose was allowed to get out, go from high to low, it should have turned colors. I'm going to check my little test strip here. Oh, sure enough, glucose got out of the beaker. In other words, the size of glucose is smaller than those pores. I'm Roy Treveline. This is BioBites.